I would like to introduce Professor Ariel Revel, medical doctor of obstetrics, gynecology and fertility, and director of medical affairs at Kadima STEM. Professor Revel, please.
I will stick to English. I understand that some people are English speakers. Can I have my next slide, please? So first, I would like to thank the organizing committee for allowing us to present our data. Okay, so just to keep you a little awake, does anybody know how many cells do we have in our body? Just a guess. 3 billion. How many? 3 billion. Billion. Okay, 3 billion. So, the correct answer is that we have 30 trillion cells in our body, but we have 40 trillion bacteria, which is the basis for a completely different lecture that I give another occasion about the microbiome. And many cells are the red blood cells, but we have all kinds of types of cells. Of course, the sperm cell and the egg cell, and we'll be talking a little about nerve cell. But each cell type performs a diverse and unique function, and disruption of our cells leads to disease. So myself, I'm a trained professor of obstetrics and gynecology. I work in vitro fertilization and make babies, but a few years ago I joined a company which I'm talking to you today about, which is involved in cells. And we start from blastocyst, and this is a blastocyst that was donated by a couple who completed their family plans. And from this we have the possibility to make all kind of cells, because we're talking about multipotent cells, and they can make lineages of all the types of cells that we have. And I would like you just to, for a moment in this time of the afternoon, to imagine a world where we have this technology, let's say 100 years from now, that cell therapy is a reality, and we have the possibility to make whatever cell our body needs. And in my dream, at first it will be for disease, but since I give lots of lectures about longevity and about lifestyle and about how to keep healthy, and I try to convince people about all kinds of issues that have to do with their personal health, I think that in the future people will come for regular treatment to replenish cells for health, not only for disease. But our vision at Kadima STEM is that we are replacing malfunctioning cells in degenerative disease, and we transplant healthy and functional cells. And currently we have 2 lines of research. One is dealing with our product, which is called AstroRx, which are astrocytes, and the other one is IletRx, which are cells that are able to treat diabetes. And we have 3 major challenges when we do cell therapy. The first one is to do no harm, primum non nocere. Then we want to make sure that the cells that we make do what they're supposed to do. And the major challenge that we are facing currently is that they are protected from the immune system. So, as our previous speakers have explained, we have steps in cell therapy product development. There is the early preclinical and then advanced preclinical. And then we have phase 1, phase 2, phase 3, and until it is approved, there are, either of our projects is at a different step, and I'll tell you where it is. Everything I tell you right now is patent protected, and lots of the data you can find, of course, in our publication and our website. So the first line we're going to talk about is AstroRx, and this is for amyotrophic lateral sclerosis program. So I don't need to tell this audience too much what is amyotrophic lateral sclerosis. In the United States, it's called Lou Gehrig disease. And there are quite famous people who are ill with this disease. And we chose this disease not because it is so unique, but because of perhaps it is easier for people to realize that we will be able to treat diseases with astrocytes. And the other project, which I think is currently a lot of our focus at the company, is about type 1 diabetes, and this is certainly a huge, huge market and very different. So this is one astrocyte. Of course, it is stained with, but we can see that it is a cell with lots of arms, and its purpose is to treat the neuron, to keep maintenance of the neuron that it is normal. And here you can see how it looks, how a normal neuron and astrocyte around it look. And this is in sporadic ALS and in familial ALS. And you see that the major issue is that the neuron has astrocytes around it that are supposed to keep it healthy. And in ALS, there is aberrant, and it is secreting toxic factors and inflammatory mediators. And when we look at the possibility to treat this disease, we need to address these issues. So this is a representation of the brain, a healthy brain, versus a brain of a neurodegenerative disease. And that, as I've said, there is in the PubMed list of many neurodegenerative diseases. Amyotrophic lateral sclerosis is just one of them, but there is no reason to consider that astrocytes will not be useful for other neurodegenerative diseases. So when we started this project, we published our results first following intrathecal transplantation of our cells into an animal model, and we saw that we have the possibility to have those rats which run on a treadmill, and they have a model that is similar to ALS, and we saw that they stay longer. The time to fall from the rotarod when we injected them with the AstroRx was longer, so they were healthier, and grip strength, which is a different test for the rats, has shown that these rats that have the ALS or the neurodegenerative model injected with the astrocytes have better strength. And from this, we have the possibility to do a clinical trial. So we start with the embryonic stem cells. We make the astrocyte progenitor, and now currently we have a frozen product that is a shelf product that we can freeze at our manufacturing, ship it to the hospital, thaw it, and inject it to the CSF of the patient to try to improve the study. So this is the first study which we conducted at Hadassah Medical Center in Jerusalem. There were cohort A, 5 patients which received a single intrathecal administration of 100 million astrocyte cells, and the cohort B received a single administration of 250 million astrocyte cells. It was planned that the cohort C and D will receive 2 administrations 3 months apart, but when we completed the first 10 patients and we were supposed to recruit group C and group D, the disease, the COVID started, and the department was converted to treat COVID patients, so we could not complete this trial. So from this, I will move to the next product, which is the potential to cure type 1 diabetes using islet cells. And this is how, islet cell transplantation is already functioning. It is taken from cadaveric. But there is a very limited limitation because there is lots of lack of donors, and they need long life suppression. And our solution is to make, in a few steps, endocrine islet cells, which we call islet RX. And after a 30-day production, we have these cells which are looking similar to islet cells. And when we inject them into mice, you can see that they keep the level of glucose normal, and we have mice that are a few months with this. We have a patent on selection of these cells so that the selected cells do better than those that are not selected. And we can, by this way, have the cells that are functioning better. Currently, our main goal is to find a way to protect our cells, because in the ALS, we inject it intrathecally, so it is in an area that is immune privileged, so we don't need to protect our cells. They don't need to face the immune system. But for diabetes, we transplant it intrathecal, and right now we have a very interesting collaboration with a company called iTolerance here in this picture. Currently we have this gel which has a fast ligand which is supposed to help our cells and protect them from the immune system. And this company in Miami currently is transplanting our cells with the gel with a fast ligand to protect them from the immune system. And we are currently following a first meeting with FDA together with them, facing pre-IND towards a clinical trial, which we hope will happen sometimes in 2 years from now. So to summarize, we have this purification system. It improves safety profile, and currently this grant with iTolerance enables us to go ahead for pre-IND. In the last minute of my presentation, I want to talk about the future of regenerative medicine, which I think is going to be a multidisciplinary field, including cell biology, genetic, biomechanics, material science, computer science, and the ultimate target of which is returning normal function to defective cells and tissues. And there's a huge global market for this in the future. And I think if you look at some animals, this whale lives for 300 years. Why can't we live more than our 90 or 100 years? There is mainly the limitation that our cells deteriorate. And I think that in the future, regenerative medicine, which already is a category in the FDA, will be the solution. This is an immortal jellyfish, and it has a secret that it is enabling. It has a possibility of changing one cell into another type of cell. So as humans, as mammals, we don't have this capacity, but maybe with cell therapy, we will be able to replenish cells that are old and dying, and we'll perhaps will be able to live healthier and longer life. So thank you for your attention. I would like to thank the people that are leading this company. First and for all, my father, Michel Revel, who started this company in the first place, Asaf Shiloni, our CEO, Ronen Tuito, our chairman, and Arik and Kfir is in charge of the iLet RX program, and Michal of the AstroRx, and thank you for your attention.